and O. So, 4 types of blood groups we are observing. And here, the blood groups are represented by a gene, that is I. I is the gene which is representing the blood group. And here, A and B, if you are taking, they are having a special kind of sugar, RBCs. They are producing sugar. The sugar tries to protrude out from the plasma membrane of the RBC. And here, a kind of sugar which is present, depending on that, the blood groups are named. And here, A, B, AB and O. Here capital I is representing a gene. But in case of O, it is not producing any kind of sugar. And there, the gene is represented by small I. Gene is represented by small I. And here, A, B are always dominant over O, because A and B, they are producing a particular type of sugars, but I is not producing any sugar at all. And here, different blood groups, they are having different types of genes. So, the basic is capital I and small I. Now see, if A is a blood group, it is having a possible genotype, that is capital I, A, capital I, A. It is representing blood group A. This is homozygous. And heterozygotic condition, if you are observing, it is indicated by IA and small I. So, this is a group which is representing a blood group A. Then when we are studying about the B, B is also, it is represented by IB, IB, IB and small I. It shows blood group B. Then when AB is there, that time IA and IB, it is in heterozygotic condition. So, it is representing the blood group AB. Now when O comes, O is indicated by small I and small I. So in this way, the genes you people are observing, and depending on that, you are calculating the blood groups also. And how to calculate? Already I told you people. Here, we will go to our slide now. Here, unit 3, part 3, that is genetics. Now see the first question. Usually, the recessive character is expressed only when present in a double recessive condition. However, a single recessive gene can express itself in human beings when the gene is present on. Where the gene is present. See the options. Any autosome, X chromosome of female, X chromosome of male, either on autosome or X chromosome. But here, out of these 4 options, option C is the right answer. Here they asked you about the autosomes. Okay, so autosomes and allosomes you people are knowing already, and here X chromosome of male is the right answer here. So we'll move to the next slide. Here, just see, two allelic genes are located on. Two allelic genes are located on. Option A, the same chromosomes. B, two homologous chromosomes. C, two non-homologous chromosomes. D, any two chromosomes. But here, option B is the right answer. Here, allelic genes are located on two homologous chromosomes. So, this is the right answer. Option B is the correct one. Move to the next. Now here, in human beings, the color of skin is controlled by. Here, some of the genes are responsible to responsible for controlling the skin color. And here, just see, multiple alleles, lethal alleles, polygenic effect, then none of these. Here, the right answer is multiple alleles. I explained to you earlier what is multiple allele. Multiple alleles are those, or you may say like this, 3 or more than 3 alleles present on a particular locus of homologous chromosome. So, compare this here. In human beings, the color of a skin is controlled by multiple alleles, that is option A. So, we'll move to the next one. See here, which of the following is genetically dominant in man? Which is genetically dominant in man? See the options. Color blindness, Rh positive, hemophilia, then albinism. So here albinism, hemophilia and color blindness, what you people are observing, these are the disorders. And here Rh positive, usually for this the right answer is Rh positive, that is rhesus positive. Rhesus, it is extracted from the rhesus monkeys. And there the same character is observed in human beings also. And maximum human beings, they are having Rh positive. So the right answer is option B, Rh positive. We'll move to the next question. Which of the following is an inherited trait in man? Inherited character in man. Trait is nothing but a character. So, we'll see here the options. FSH, follicle stimulating hormone. LH, luteinizing hormone. TSH, and finally that is the RH. RH is rhesus factor. And actually, inheritance of rhesus factor you people are observing in man. So, option D is the right answer, that is RH. We'll move to the next. At what temperature the blood is stored in bottles? So, the blood is to be stored in the bottle, you people are knowing. So, here, at what temperature you are storing? Just see here the options. 4 degrees centigrade, 37 degrees centigrade, 0 degrees centigrade or 25 degrees centigrade. Yeah, the right answer is option A, 4 degrees centigrade. So here the blood can be stored comfortably. We'll move to the next slide. Here, in how much maximum period the stored blood should be transfused? Now you are storing blood in the blood bank. How many days you are storing it? Okay, so and after that when it is to be transferred or transfused. Now see the options: 7 days, 10 days, 1 month, 15 days. Here, 1 month is the right answer. So, the maximum period of the stored blood should be transfused 1 month is the right answer. So, option C is the right answer. We'll move to the next. Now, the stored blood must be free from. Now, the blood you are storing, it must be free from all the diseases. Okay, it must be a pure blood. So, we'll go to the options. Option A, impurities only. Option B, viruses only. Option C, infectious biotic potentialities. Last, D, bacteria only. So, no infectious material should be there in the blood. It must be very pure. So, option C is the right answer. Here, infectious biotic potentialities is the right answer. So, option C is the correct one. We'll move to the next. Now, to store blood, some anticoagulant is added. It can be. What anticoagulant is added? We'll go to the options. Sodium chloride, sodium oxalate, potassium chloride, thromboplastin. Here, the option B is the right answer, that the anticoagulant is added, that is nothing but sodium oxalate. So, sodium oxalate is the right answer. We'll move to the next. Now see. Sir, can you minimize? If a certain patient with blood group B requires immediate blood transfusion, the following type can be given to him. What following type is followed there? Just see the options. O and B, O and AB, A and AB, B and AB. Yeah, the right answer is option A, that is O and B. You people are knowing blood groups, which blood groups they are producing sugar and which genes they are not producing sugar. Everything you are knowing. Depending on that, if you calculate, then you will come to know that is option A, O and B is the right answer. We'll move to the next. Here, inheritance of ABO blood group system is an example of. That is, inheritance of ABO blood group system is an example of. The 4 different options are here. Just see. Multiple allelism, partial dominance, epistasis, then dominance. Here the right answer is multiple allelism. Already I explained. That is the multiple allelism. Option A. Next. A person with the blood group A can be given blood of which group? Blood group A is there. Okay, the blood is to be given to him. Which blood group you are suggesting or you are giving? Just see the options. A and B, B and O, A and O, A, B, AB and O. So here option C, A and O you can give. O is a universal donor, A as it is. So both you can give it to him. And you are knowing, if suppose the blood which is not matching, if you are giving, then clumping of blood you people are observing. And clumping, it results into death of the patient. So as a result, you have to be very particular regarding these blood groups. Here, option C, A and B, A and O is the right answer. So we'll move to the next. Anti-serum used to detect RH blood group. Anti-serum used to detect RH blood group. So, anti-A, anti-B, anti-C, anti-D. Anti in the sense antigen A, antigen B, antigen C, antigen D. So here the right option is D, that is antigen D. Here to detect RH, antigen or anti-D you people are used. Next, here who was the scientist who introduced A, B, O blood groups? The name of the scientist you are mentioned. Here, Weiner, Levine, Fisher, Landsteiner. So here, option D, Landsteiner is the right answer. So here, ABO blood group was suggested or introduced by Landsteiner. Next one. Now see, during serological test, in which anti-human serum is mixed with blood of another animal. Blood of which animal gives the thickest precipitate. Here, which animal you are using for the experimental purpose? One is gibbon, chimpanzee, dog and mule. So here, the right answer is option B, chimpanzee's blood is used. So, next one. Matching among close relations is referred. So, close relation you are observing. Here, permanent marriage, line breeding, inbreeding, crossbreeding. What it is indicating? Mating among close relations is referred. It is referred to option C, inbreeding. So, inbreeding process here is referred here. So, next one. If one parent belongs to A blood group and the other belongs to O blood group, their children possibly represent A and O blood group. Okay, here what we will do? I am calculating this in front of you, then you will come to know. One is A, another is O. Okay, now we will see. We will calculate and see here. So, A and O. A blood group is crossed with O blood group. Now, here you want to see a possible blood groups, then you have to take it as in heterozygous condition. So, here genotypes you can take IA and I. They are becoming heterozygous genotypes. Here you cannot take heterozygous, O is having small I, so as a result it is homozygous in nature, that is I and I. Now, these genes they are producing after cross. They are in the beginning they are producing gametes. So here two types of gametes are produced, IA and small I. And here as it is homozygous, it is producing only one type of gamete, that is I. Now, when crossing is made, just see, here the combination is capital IA and small I. Another combination is small I and small I. So, possible blood groups are A and O. Just go to the slide and just see to it. Here, if one parent belongs to A blood group and other belongs to O blood group, the children possibly represent here option C, A and O groups only. Okay, here in O blood groups you people are observing. Okay, so you calculated A and O. Just see here, you got A and O. So option C, A and O blood group is a right answer. We'll move to the next. Just see. The next one. The chromosomes responsible for the determination of sex are called. The chromosomes which are responsible for the determination of the sex. Okay. So, they are called, just see, autosomes, allosomes, multicolorians, then heterosis. Here the right answer is allosomes. Option B. Autosomes and allosomes. Already I explained. So the right answer is option B. Now, who put forward the theory of linkage? Who has given theory of linkage? Just see here. Sutton, Morgan, DeVaris, Bateson and Punnett. Here, actually, the theory of linkage was given by Morgan. So, as a result, option B, Morgan is the right answer. Move to the next. The bar body is observed in. Where you are observing the bar bodies? Just see. Basophils of males, neutrophils of females, eosinophils, then neutrophils of males. The right answer is option B, neutrophils of females. Next, mule is an offspring of. The mule you people are knowing. Okay, it can go fast, it can climb high, as well as it can carry a weight. Okay, so what is the combination resulted into mule? Just see the options: male and female donkey, cow and ox, male ass and mare. The last one is male horse and female ass. The right answer is option C, male ass and male. Male ass and male combined together to form the mule, which is very popular now. Next one. The genotype of a boy having sexual characters of a girl is. Boy is having girl character. Now see the option XX, XXY, XO, XYY. The right answer is XXY. Here, the genotype of boy having sexual characters of girl. Okay, here what happens? The characters will be changed because of the trisomic condition. So here, one addition the chromosome the person is getting, that is the Y chromosome. So X, X and Y. This is the right answer, that is option B. Next. The average ratio of male to female individuals based on XX and XY type of sex determination in total world of human population is. Okay, the right answer just see 3:1, 1:3, 1:4, 1:1. The right answer is option B, 1:1. So 1:1 is the right answer. We'll move to the next. The sex determination pattern in honeybee is called, sex determination in honeybee is called, just see, female haploidy, haplodiploidy, gametic diploidy, gametogony. So here the right answer is haplodiploidy. Option B, haplodiploidy is the right answer. We'll move to the next. Now see, sex of human child is determined by, sex of the human child, it is determined by, see the options: size of the egg at the time of fertilization, size of the sperm at the time of fertilization, sex chromosomes of father, sex chromosomes of mother. So the sex of the human child is determined by the sex chromosome of the father. Father decides it. So we'll move to the next. Option C is the right answer here. Now, Leon hypothesis deals with, Leon hypothesis it is dealing with, just see, centromere position, genetic compatibility, then genetic incompatibility, number of bar bodies. It is totally, the Lian hypothesis is dealing with number of bar bodies. So the right answer is option D, number of bar bodies. Next one. Animal which remains male initially, then changes to female is called, that is example, tapeworm, proglottides. Now here the options are protandrous, apomixis, profixation, none of these. So the right answer is protandrous. Here, protandrous type of animals you people are observing. So, friends, we'll stop this clip here. The same will continue in the coming class. So, thank you very much.